Hello, this is ARYANA.
And Dornaz.
We are going to talk about how temperature affects the Beta Vulgaris cell membrane permeability. Our presentation is going to be presented in 3 parts, and they are hypothesis, techniques, and result. So now I'm going to talk about the hypothesis. As you know, cell membrane is made by phospholipids, protein, and carbohydrates. And also, phospholipids are amphipathic, as hydrophilic head and hydrophobic tail. And also, cell membrane fluidity and permeability depend on phospholipids and cholesterols. So, the relation between the characteristics of the cell, which are the fluidity and the permeability, bring up the hypothesis of if the lipid composition of the cell membrane is manipulated by changing the temperature and the Beta Vulgaris membrane fluidity is affected, then variations in cell membrane permeability may be observed in response to the temperature fluctuations. And now I'm going to talk about the techniques. So first, we're going to take some bead samples, and then we're going to cut them in same sizes. It's better if you carefully cut them in the same size because it's going to affect the future measurements. In the next step, we're going to fill distilled water in 6 test tubes, and we're going to put them aside with 10 milliliter. So the 6 test tubes are going to be with different temperatures. The first one is the 0 degree, the second one is the room temperature, which in our project was 17 degree. The third one is the 35 degree, the fourth one was 50 degree, the fifth one was 65 degree, and the last one was 19th degree.
Thank you, ARYANA. After preparing the test tubes, we put them into water bath. But before that, we set up the water bath on 35 degrees, 50 degrees, 65 degrees, and 90 degrees. And beside of that, we set up an ice bath for 0 degree. After 30 minutes, we poured the sample solutions into cuvettes, and as you can see, there is variety of coloring and the released pigment. This is room temperature, which was 17 degrees. This was 19 degrees, and as you can see, the color changed. This is 0 degree, 35 degrees, 50 degrees, and 65 degrees. After collecting the solutions into cuvettes, you should calibrate the spectrophotometer on 535 nanometer and then collect your raw data. After that, we came up with our results, and as you can see, the blue trend shows the mean values for trial 1. The pink trend shows the mean values for trial 2, and the green trend shows the mean value for the whole experiment. And needless to say, each trial had 3 replicates. And as you can see, there are 2 trends in this graph. One is upward and the other one is downward. The upward supports the hypothesis, but this downward is not rejecting it, and it's due to degradation of betaline, which is the pigment of beads. And all in all, the mean value trend supports the hypothesis. And that's all. Thank you for your attention.